It's Biomed Cafe menu. Meiosis I will be presented today. We will talk about meiosis, meiosis function, meiosis I, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Meiosis discovery. They saw something others didn't see. Meiosis was discovered for the first time in 1876 by Oscar Verfecht. It was described again in 1883 at the level of chromosomes by Edward van Beneden. The significance of meiosis for reproduction and inheritance was described only in 1819 by August Weismann. In 1911, the Thomas Hunt Morgan detected crossovers in meiosis. The term meiosis was introduced to biology by J.B. Farmer and J.E.S. Moore in 1905. Meiosis. Meiosis is a type of cell division that results in 4 haploid daughter cells, gametes, such as egg and sperm, each with half the number of chromosomes of the parent cell. Meiosis function. Meiosis function is production of gametes. Gametes, or sex cells or sperm and egg, join in fertilization, and the 2 haploid sets of chromosomes form a complete diploid set, a new genome. Interphase. Before entering meiosis I, a cell must first go through interphase. The interphase, or G1 plus S plus G2, is a much longer phase of the cell cycle than cell division, or meiosis plus cytokinesis. The cell grows during G1 phase, copies all of its chromosomes during S phase, and prepares for division during G2 phase. Phases of meiosis. Meiosis has 2 parts, meiosis I and meiosis II. Meiosis I can be divided into 4 phases. Meiosis I can be divided into 4 phases: Prophase I, Metaphase I, Anaphase I, and Telophase I. Meiosis II can be divided into 4 phases too: Prophase II, Metaphase II, Anaphase II, and Telophase II. Meiosis I. Prophase is similar to pancake, and P is their common first letter. Prophase I. In Prophase I, the cell starts to break down some structures and build others up. Each chromosome carefully aligns with its homologous partner. Prophase I events. Number 1. The chromosomes start to condense, making them easier to pull apart later on. Number 2. The spindle begins to form. The spindle organizes the chromosomes and moves them around during meiosis. The spindle grows between the centrosomes as they move apart. Plant cells have microtubule organizing center instead centrosome that plays the similar role. Number 3. 2 chromosomes match up at corresponding positions along their full length. This process, in which homologous chromosomes trade parts, is called crossing over. It helps align by a protein structure called the synaptonemal complex that holds the homologs together. Metaphase is similar to marshmallow cake, and M is the common first letter. Metaphase I. In Metaphase I, the homologous pairs of chromosomes line up on the metaphase plate near the center of the cell. Metaphase I events. Number 1. The spindle begins to capture chromosomes and move them towards the center of the cell or metaphase plate. Number 2. Homologous pair chromosomes line up at the metaphase plate for separation. Anaphase. Anaphase is similar to apple pie, and A is their common first letter. Anaphase I. In Anaphase I, the homologs are pulled apart and move apart to opposite ends of the cell. Anaphase I events. Number 1. The homologous chromosomes are now pulled towards the centriole at each end of the cell. Number 2. The sister chromatids of each chromosome remain attached to one another and don't come apart. Telophase. Telophase is similar to tart, and T is their common first letter. Telophase I. In telophase I, the chromosomes arrive at opposite poles of the cell. Telophase I events. Number 1. The chromosomes are pulled completely apart. Number 2. New nuclear envelopes form. Cytokinesis. Cytokinesis. The plasma membrane is separated by cytokinesis and two new cells are effectively formed. Cytokinesis usually occurs at the same time as telophase I. Cytokinesis result: two new cells or haploid daughter cells. At the beginning of meiosis, a human cell has 46 chromosomes, or 23 paired chromosomes. Each chromosome consists of two sister chromatids. During meiosis I, the cell undergoes reduction division and the number of chromosomes is halved. Each daughter cell at the end of meiosis I has 23 chromosomes, but each chromosome still consists of two sister chromatids. One chromosome has two chromatids, therefore 23 chromosomes means 46 chromatids. Therefore, at the end of meiosis I, each daughter cell would have 46 chromatids. Key points of meiosis. Meiosis in humans is a division process that takes us from a diploid cell, one with two sets of chromosomes, to haploid cells, ones with a single set of chromosomes. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time to the quiz game. I've put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video, bye!